Hello everyone, warm welcome to this part 4th, chapter 6, plant anatomy of flowering plant, unit 2. Here we have to discuss that is vascular tissue system, different type of vascular tissue system. Then we have to see anatomy of dicot and monocot root, what are the differences in between. Then we have to see anatomy of dicot and monocot stem. This is part 4th, chapter 6. Plant anatomy of flowering plant. Hello, let us concentrate on part 4 of chapter 6, anatomy of flowering plant. Already we have done epidermal tissue system and that is ground tissue system. Now we will start from that is vascular tissue system. I told you already, vascular tissue system, it consists of that is vascular tissue, that is xylem and that is phloem, meant for conduction of water and minerals and of organic solutes. Vascular bundle present in the stellar part that will make the vascular tissue system, and vascular bundles may be of two types. One is that is radial and one is that is conjoint vascular bundle. As I told you earlier also, radial these are found in case of root and the conjoint these are found in case of stem. More detail we'll see right now. What we mean by that is radial vascular bundle. You see, radial vascular bundle means what? The arrangement of xylem and phloem, these are on different radii. If you have cut a transverse section like this of root, you see this one is that is xylem and here this one is that is phloem. So you see, xylem and phloem, these are present on different radii. So this vascular bundle is known as radial vascular bundle. It is found in case of root, remember. Radial vascular bundle. Xylem and phloem are different radii, also known as centripetal one. This is found in case of root. One more thing, this radial vascular bundle, what are you seeing here? That if this is the xylem, this one is protoxylem and this one is, what is this? This one is that is metaxylem. Can you recall? That is protoxylem towards periphery. Therefore, the vascular bundle will be known as that is exarch. Is it right? So you have to concentrate now. In root, the vascular bundle is radial, then the xylem will be exarch here. Okay. Come to the next, conjoint vascular bundle. Xylem and phloem, these are combined. These are found on the same, that is radius. These are jointed together. Conjoint present in the same vascular bundle and on the same radius as is found in case of stem and leaves. You can see here that is like this, that this is that is transverse section and here this is the vascular bundle. We can enlarge this vascular bundle, that is the xylem and phloem. Phloem is towards periphery, xylem is towards that is center here, and these are jointed together, will be known as conjoint vascular bundle. What are you seeing here? Here this one is the protoxylem. Tell me, protoxylem is towards center and the metaxylem is towards, that is this one is the xylem with wider vessels, metaxylem is towards periphery. So here the xylem will be known as what? Endarch or centrifugal. So remember, it is found in case of stem. In stem the vascular bundle is conjoint, then the xylem will be endarch. Remember, so here it is found in case of that is stem. Is it right? One more thing we have here. You are seeing here in between phloem and xylem, the cambium is absent. As in case of monocot, the vascular bundle is known as that is closed vascular bundle. Cambium is absent. If the cambium is present, the vascular bundle is known as open one. Come, that is endarch is also known as centrifugal one. Open means when the cambium is present, as in case of dicot stem. In case of that is in dicot root also we will use the term closed when the cambium is absent, as is found in case of monocot stem. Is it right? Come to the next now. Now we'll go to the most interesting and colorful part of this chapter. We have to discuss anatomy of that is plant parts. You know, plant parts we have that is stem, we have that is root, then we have leaves. In the same way we have to go for dicot stem, then monocot stem. You have to see dicot root, monocot root, then we have to see dicot leaf and that is monocot leaf. Very important, interesting part this is. Come to the next now, that is here. First we have to see anatomy of that is dicot root, right? Dicot root anatomy we have to see now. Say, to see internal structure, we have to cut a thin transverse section of dicot root. You must have seen, that is when we used to prepare salad, that this is carrot. So here we have to cut a thin slice like this. Thin slices that will become like this, the thin slices, and this is known as the transverse section. That is when a cut is made perpendicular to the long axis, transverse section. In transverse section of the dicot root, we will see the following structure. As I told you, 𝕆𝕆𝕋, outermost one, that is piliferous layer. It is single layered one. Some cells, they have unicellular root hair, already we have discussed, and root hair, they will absorb the water and minerals. They will increase the surface area. The next is cortex, made up of many parenchymatous cells. You know the meaning of parenchymatous cells. The cells are like this, and they have, that is, large intercellular space. Living cells, these are plenty of intercellular space. Come to the next now, endodermis. Remember, here in case of root, that is hypodermis is altogether absent. Remember, important point. Endodermis, innermost layer of the cortex, it surrounds the stele. Everything inside the endodermis is known as stele. It is made up of single layer. Barrel shaped cells are there, already I told you. The tangential and the radial wall of the endodermal cells, they have deposition of water impermeable substance, a waxy natural substance, and that is suberin in the form of Casparian strip. You can see here what we mean by, yes, this is, I can make it here, that these are the endodermal cells. So these endodermal cells, on their radial and the tangential wall, they have this thickening. That thickening is known as, that is, Casparian thickening, right? And in between, that is endodermal cells, just in front of protoxylem, the endodermal cells, these are thin wall, and these thin wall are known as, these are called, that is, passage cells. These are called passage cells, okay? Just below the endodermis, there is the thin layer, that is pericycle. It is made up of thick walled, that is parenchymatous one, and the lateral, important point, lateral root originate from pericycle. Vascular tissue, as I told you right now, vascular bundles are radial, exarch. Vascular bundles vary from 2 to 6. There may be 2 to 6, maybe 4, maybe 6, known as diarch, 2 tetrarch or exarch. In between xylem and phloem, that is, patches of parenchymatous cells are present, known as conjunctiva. You can see here that this is the section here, that is here. We have, this is the xylem we have here like this. This is the xylem, that this one is the phloem we have here, and in between xylem and phloem, we have, these are the cells, parenchymatous one, which are known as, that is, conjunctive tissue in between xylem and phloem. Is it? This one is phloem, right? And this one is xylem. Okay. You can see the diagram here, dicot root. Good diagram we have here. That is, these are the unicellular, that is root hair. Unicellular these are. This is the piliferous layer, or that is 𝕆𝕆𝕋 also known as. It is called piliferous 𝕆𝕆𝕋. This is cortex. You can see the diagram here. And this one is endodermis. Below endodermis there is pericycle, and these are the xylem. This one is the phloem. You can see this outline sketch, ground plan. Then the cellular sketch you can see here. These are the root hair. Root hair, you know, these are that unicellular one. This is epidermis. This one is cortex. This is made up of thin walled living cells. And this is the endodermis one. What are these? That is, this one is the xylem. In xylem, what is this? This one is the, this is the protoxylem. Is it? You can remember protoxylem. And this one is the metaxylem. Therefore, this is, condition is exarch. This is phloem. This is xylem. Number of the vascular bundles are 4. That is known as tetrarch. Okay. Now you have to see almost similar structure we have in case of monocot root also. You see, in monocot root also, the outermost layer is, that is 𝕆𝕆𝕋. That is, some of the 𝕆𝕆𝕋 cells they have, that is unicellular root hairs are there. The cortex is there, as usual cortex. Now this is the endodermis is there. This one is, that is endodermis, you know. Endodermis, it is having what? Casparian strips are there, visible. And it has, see the difference here. Here the vascular bundles were only that is 4 patches are there. Here we have more than 4 patches. That is one xylem, one phloem will make one vascular bundle, and in that way it is having many vascular bundles. And metaxylem is towards center, protoxylem is towards periphery. Therefore, the vascular bundle as usual that will be what? Exarch 1. You see here in this case the pith is usually absent. Pith is absent here, but in this case this is the tissue. The pith is very well developed. This is the pith. This is TS of monocot, right? This is TS of monocot root. Come to the now, that is anatomy of monocot root. As I told you in diagram also, it is almost similar to that of dicot root in many respects. It has epidermis. Better to call epidermis, we can call that this one is, that is 𝕆𝕋𝔼. This is, and from 𝕆𝕆 there will be what? That is root hair, unicell root hair. The cortex is there, endodermis is there, pericycle is there, vascular bundles and pith is there. But as compared to the dicot, some of the differences are there. That is, in case of dicot root, we have few, that is xylem bundles, few vascular bundles. There are usually more than 6, as I told you in diagram also. You can see here. Here there are 4 to 6. Here the vascular bundles are, that is, many vascular bundles are there, right? So you see, pith is large. You can see once more here. Here pith is absent, or if it is present, that is very, very small one. But here the pith is larger. Pith is there in case of, that is, monocot root. Large pith is there. Is it? Monocotyledon roots, they do not go under secondary growth. That's why these are thin. While in case of dicot root, you have to say that is dicot root, secondary growth is present. So we can see it now. Difference between, that is, monocot and dicot root. Can you summarize it now? Yes, come on. Pericycle, in case of monocot root, it produces lateral root. Yes, in dicot also, very important point this one is, in dicot also, that is, lateral roots are produced. You can see here that this is the, that is, root, transverse section of root. This is the endodermis, and below endodermis this one is, that is, this is the pericycle. What is this one is? That is pericycle. From pericycle the lateral roots come out from pericycle. Remember, lateral roots come out from pericycle in both monocot as well as dicot one. But in case of dicot root, this pericycle, beside making lateral roots, from this vascular cambium and cork cambium also develops. The detail of this we will see when we deal with, that is, secondary growth. So here pericycle is having 3 functions: lateral root, vascular cambium and formation of cork cambium. Is it right? Vascular bundles, polyarch, many, numerous, while here dia 2 hexars, 2 to 6. Cambium is absent, altogether absent. But important point this one is, in dicot also, you see that this is the phloem, that this is the xylem, and that this one is the phloem. That is, in between xylem and phloem, the cambium is not present since beginning, but it appears at the time of secondary growth. Remember this thing. Pith, can you compare pith? Pith is small or absent. Pith is large in case of monocot root. You see here, yes, that is, pith is large and well developed, while this is small or that is absent in case of, that is, dicot root. Okay, come to the next now. We have to see now, that is, anatomy of dicot stem. Yes, dicot stem. This is, we can write it, anatomy of dicot stem. Then we have to compare it with monocot stem also. Epidermis, when we have discussed epidermal tissue system, I told you, in case of, that is, dicot stem, these are the epidermal cells. These are the living parenchymatous cells making a continuous layer. Some of the epidermal cells, they will make this multicellular hairs, trichome, and these are, because this is the aerial part, these are surrounded by, that is, vivaxy layer known as, that is, cuticle. And what are these? These are the, that is, trichome. Is it right? This is the outermost epidermis. Come on. In transverse section, typical of young dicot stem, that is epidermis is the outermost protective layer. It is covered with thin layer of cuticle, number 2 point, and it may have, that is, several multicellular trichomes. In green stem, young stem, the stomata may also be present. These are the stomata here for gaseous exchange. Second one, cortex. The cell arranged in multiple layers. You can see the diagram first. That this is the epidermal hair. These epidermal hair, these are, these are, that is multicellular, these are. Then this one is epidermis. Now what is this? That is, here we have, just below the epidermis we have, that is hypodermis. Is it? Hypodermis. Below hypodermis we have, that is, this is parenchymatous cortex. Then there is endodermis. Then there is pericycle. We have to see. You can see it here. You can compare it like this. I'm going to make this diagram. This is outer sketch. This is the epidermis one. And epidermis is having that these are the, that is multicellular shoot hair. That is, below this we have, what is this? Below this we have, this one is, that is the hypodermis. This one is, that is hypodermis we have here. Is it right? This one is the hypodermis. Then here we have, this is the endodermis. So in between endodermis and hypodermis, that is, here we have, this is the cortex, and this one is the, this one is the endodermis. Is it right? This one is endodermis, this one is the cortex, and this one is the, that is hypodermis. This is 𝕍𝕆𝕋𝔼𝕆 hypodermis, this one is, and this is parenchymatous cortex. Inside this, we will have, that is pericycle, and we have, these are the vascular bundles like this. These are the vascular bundles. You see it first. Vascular bundles, can you summarize? What is this? This one is phloem outside, and this one is, that is xylem inside, and protoxylem is inside. Therefore, the xylem will be here, but xylem will be enlarged. Are you getting my point? Am I clear? Yes, we have to see detail of this now. On this basis, we have to, that is, learn now. Cortex just below, that is epidermis, we have cortex. Arrange multiple layers between epidermis and pericycle, that is known as cortex. Is it? That is, cortex consists 3 zones. Just below the epidermis, this is hypodermis. Hypodermis, it is 𝕍𝕆𝕋𝔼𝕆 one. You see here, this is epidermis. This one is hypodermis. This one is endodermis we are talking about. So here this hypodermis, this is 𝕍𝕆𝕋𝔼𝕆 one, that is, which provide mechanical strength. This is the cortical region made up of so many, that is parenchymatous cells. Layers are there. Cortical layers below the hypodermis consist of round, thin-walled parenchymatous cells which have large intercellular space, and this one is the endodermis, innermost layer of the cortex, endodermis. The cells of the endodermis, these are rich in starch grains, therefore it is known as starch sheath. Is it right? And below this endodermis, we have the pericycle. Pericycle, either it may be in form of continuous layer, or it may be in the form of, that is, patches also. Pericycle is present on inner side of the endodermis, and above the phloem, it is in the form of semilunar patches. I am going to make one vascular bundle here. That is this vascular bundle. This vascular bundle, what is this? This one is the phloem. When the pericycle is in the form of patch, so this is found, that is just above the phloem. This is phloem. This one is xylem. Semilunar patches are there of sclerenchyma. In between the vascular bundle, there are few layers of radially placed parenchymatous cells which constitute the medullary rays. You see that these are the vascular bundle and these are the parenchymatous cells which will make the medullary rays. Is it clear? Come to the next now. We have here, that is vascular bundles. In case of dicot stem, I told you vascular bundle, that is these vascular bundles, you see, vascular bundles are like this. Vascular bundles, these are arranged in rings. If this is the endodermis we have here, then these are arranged in ring. That is, number of vascular bundle is not much more. Large number of vascular bundles are arranged in a ring. Arrangement of vascular bundle. Characteristic found in dicot root. Each vascular bundle is conjoined. This is phloem. This one is xylem. In between this there is, that is cambium is there. That's why the vascular bundle, you can correct it, that the endodermis will go like this. Okay, and this is the cambium we have here in between. This is the cambium. So therefore the vascular bundles are conjoint, open, with enlarged xylem. This is cambium, phloem is here, and protoxylem is towards the center, towards the pith. That's why this is enlarged. Large number of rounded parenchymatous cells with intercellular space, they will occupy the central place. Here this one is the pith. Come on, this is the pith, this is the medullary ray, and this is the vascular bundle, that is conjoint. Then this is with enlarged, this is pericycle, this is endodermis, this one is parenchymatous cortex, this is colon-chymatous hypodermis, this is epidermis, and this is the epidermal hair. This is a part of this we have magnified. Let this part, we have magnified it like this. So here these are the hairs. This is epidermis, this is the hair, this is colon-chymatous hypodermis. Are you getting my point? This one is parenchyma. Here this one is endodermis, this is endodermis, this is pericycle patch, this one is phloem. We can make it like this. Let this is the phloem like this. This is the cambium we have here. This is metaxylem. This one is protoxylem. Therefore the vascular bundle, that is xylem, is, that is enlarged, and vascular bundle is, that one is conjoint. Am I right? Okay, clear? Come to the next now. We have to see now anatomy of monocot stem. Monocot stem, little different. Monocot stem, the sclerenchymatous hypodermis is there, that is epidermis. Is it right? This is the epidermis we have here. Epidermis is having cuticle outside. Usually the trichomes are absent. That's why the surface is smooth here. Here below we have sclerenchymatous, that is hypodermis, and rest of the tissue, that is ground tissue system, is undifferentiated. A large conspicuous parenchymatous ground tissues present, which is undifferentiated, means we do not have xernal cortex, we do not have endodermis, we do not have pericycle, and we do not have pith and medullary rays. All these are absent. Ground tissue is undifferentiated. Important point. Now in this ground tissue, number of vascular bundles are found. Vascular bundle, I am making here one vascular bundle. This is enlarged. All vascular bundles will be enlarged because this is stem. You see, enlarged means protoxylem is towards inside. I am going to make here. So there are so many vascular bundles you are seeing here, and these vascular bundles, the larger vascular bundles are towards the center, like this you are seeing, and the smaller vascular bundle, these are towards the periphery. So there are numerous vascular bundles, not arranged in a ring. These are found scattered. Large number of the scattered vascular bundle. Each is surrounded by sclerenchymatous bundle sheath. This is the bundle sheath we have here. And the vascular bundle, that is, what will be this? This will be phloem, that will be xylem, that will be bundle sheath. These are scattered. That's why the vascular bundle are conjoint. Cambium is absent, closed. Peripheral vascular bundles are generally smaller, and the centrally located are, that is, larger one. Phloem parenchyma is usually absent, and here water containing capacity is there, cavity is there. You see, if I am going to make one vascular bundle here like this, that this is the center here, right? So this is the metaxylem and this is the protoxylem. Usually the xylem vessels are arranged V-shaped or Y-shaped, and near to the protoxylem, that is, a cavity is there which is known as water cavity, and here that will be the phloem will be there. Vascular bundle is conjoint, then this is enlarged, and this is water cavity we have here, that is in vascular bundle. Okay, come to this next. This is the outline sketch, epidermis. This is the hypodermis we have, which is sclerenchymatous one, you know. Vascular bundle, these are numerous you are seeing, and this is, I told you detail, ground tissue, that one is, that is undifferentiated ground tissue system. This is the part and last we have here, epidermis. Usually that hairs are absent. I told you smooth is there. Hypodermis, vascular bundle. If you see this vascular bundle, this one is metaxylem, and this one is the protoxylem. Here is lying the water cavity. Is it right? Phloem, xylem, ground tissue. Right? Thank you. This is part 4 of chapter 6. We have discussed anatomy of dipot and monocot stem. Thank you.